Assalamu alaikum. In this lecture, we will cover mitochondria and peroxisomes, which are involved in bioenergetics and metabolism. So let's start with the mitochondria. Mitochondria are thought to have evolved from bacteria, and you would notice from the structure of mitochondria that they do look like bacteria and they have bacterial features. Now, the main function of mitochondria is generation of ATP via the electron transport chain process. Now, most of the mitochondrial proteins are translated from the nuclear genome rather than their own DNA. So they do contain their own DNA, except that it only encodes for a few proteins that are involved in the process of ATP generation. So this is the basic structure of mitochondria. You would notice that there is an outer membrane, and this outer membrane is quite porous. And then you have an inner membrane right here, and this inner membrane contains it's rich with proteins, if you remember from the first lecture, and especially the complexes that are involved in the electron transport chain. Now, notice how convoluted it is. It has this distinct structure in order to contain as many proteins as possible. Okay, so in between the outer membrane and the inner membrane, you have the intermembrane space. That's where protons accumulate, ready for re-entry into the matrix, which is right in the heart of the mitochondria. Okay, and that's where metabolism of pyruvate and the Krebs cycle take place. Now, so we have the outer membrane. It's quite permeable to small molecules. It contains openings called porins. There is the inner membrane. It contains high percentage of proteins. Okay, it's formed from several folds to increase the surface area, and that's the function of the inner membrane. Okay, so mainly generation of ATP. It can also transport a number of metabolites like pyruvate. Overall, in contrast to the outer membrane, the inner membrane is impermeable to most ions and small molecules. Okay, now we talked about the intermembrane space. The matrix contains metabolic enzymes, especially those involved in the Krebs cycle. Now, mainly now, mitochondria are located in cells that require high energy. So in muscle cells, there are large number of mitochondria compared to other tissues like liver, for example. Mitochondria overall are dynamic, so they're not static. Rather, they undergo fusion with each other. They can also undergo sort of divisions, or what is called mitochondrial fission. Okay, so fusion would increase mitochondrial oxidative capacity. Fusion of different mitochondria can also repair damaged mitochondria. Okay, so fission on the other hand increases the resistance to oxidative stress, as well as segregation of damaged mitochondria. Now, and mitochondria have several functions, by the way. Okay, in addition to generation of energy, including autophagy. We talked about that in the part of vesicular transport, as well as control of cell death. Now, as I said, mitochondria have their own genetic material. They have circular DNA, just like bacteria do, and this genome can encode for 13 proteins as well as few rRNA molecules and tRNA molecules. Now the proteins that are encoded by mitochondrial genome are those involved in oxidative phosphorylation. Now, most of the mitochondrial proteins are encoded by the nuclear genome. Few proteins, as I said, are encoded by the mitochondrial genome. Okay, so you can connect this to diseases, genetic diseases, that most mitochondrial diseases are caused by mutations in the mitochondrial genome. So how are proteins imported into mitochondria? How are they inserted in the inner mitochondrial membrane or the outer mitochondrial membrane? Well, what happens is that there is a sequence known as a pre-sequence, and this sequence is possibly charged overall, and it's taken by a chaperone known as chaperone 70, heat shock protein 70. So this protein is taken into the mitochondria in the intermembrane space, and then there is a complex known as a TIM complex. It inserts, it takes bacteria, it takes these proteins into the inside the matrix of mitochondria. Okay. Now, if a protein contains a transmembrane domain, then the TIM complex also inserts the protein into the membrane. Okay. Now, so there is a pre-sequence. So how are proteins targeted into the inner membrane space? Because not all of them have a pre-sequence. Well, some of them have multiple internal import signals. Okay, and what happens is that you have chaperones again that target the proteins inside. They take the proteins inside the mitochondria. Once they get into the TIM complex, and there are several TIM complexes, they recognize that this protein must be inserted into the inner membrane. Now, notice something here. You have some also inner membrane proteins that are encoded by mitochondrial genome, and they are inserted into the inner membrane space using another channel protein known as a translocase, OXA translocase. So there are two mechanisms by which these proteins can be inserted. There are three mechanisms by which these proteins can be inserted into the inner membrane. Either that they have a pre-sequence and they also have a membrane, transmembrane domain, okay, or like in the previous slide, or that they do not have a pre-sequence, but they do have internal import signals recognized by TIM2. And the third mechanism is if a protein is encoded by the mitochondrial genome, then and if they have a transmembrane domain, then these proteins are recognized by OXA translocase and they are inserted into the inner membrane. Now, the proteins that are supposed to be located inside the outer membrane, they are recognized by another complex known as TOM complex. Okay, and many of these proteins have alpha helical transmembrane domains. Now, notice something, that there is another complex known as the SAM complex, and this complex recognizes proteins, membrane proteins, that have their transmembrane domains looking at beta sheets rather than alpha helices. Now, note something. In general, human membrane proteins have their transmembrane domains as alpha helices. Bacterial membrane proteins have their membrane domains as beta sheets. Okay, so so note the similarity between mitochondria and bacteria, whereby both have transmembrane domains of membrane proteins having a beta sheet structure rather than alpha helices. Now, mitochondria are also they have a specific type of lipids in their membranes. Now, for example, mitochondrial membranes do have phosphatidylcholine, phosphatidylethanol both of which are synthesized in the endoplasmic reticulum. Then they are carried to mitochondria by transfer proteins. Now, they also contain in their membranes phosphatidylserine, which is synthesized from phosphatidylethanol. Now, they also have a unique phospholipid known as cardiolipin, which is rich, as you can tell from the name, in cardiac tissues. Now, this lipid, we talked about it before in biochemistry. It contains 4 fatty acids, and it is synthesized in the mitochondria. This phospholipid is thought to enhance the efficiency of the oxidative phosphorylation process in mitochondria. Okay, so let's talk about mitochondrial diseases now. The thing is that we inherit our mitochondria from mothers mainly, rather than fathers. The contribution of fathers is only DNA. So any disease would be transmitted through mothers to children. Okay. So you can refer to this website, and this website has a nice categorization of mitochondrial diseases. So basically, we can classify these diseases according to what causes them. So they can be classified as genetic diseases or biochemical diseases. So the biochemical classification of mitochondrial diseases is the following. So basically, you can have the 5 categories for these diseases, depending on the location of the defect. So you can have a problem with the transport of substrate into the mitochondria. There could be a defect in the utilization of substrates, or you can have defects in any one of the enzymes involved in the Krebs cycle. In addition, there are 4 complexes involved in the respiratory chain reaction, and mutations can also exist in any one of these complexes. Finally, there could be a problem in the generation of ATP via the ATP synthase through oxidative phosphorylation. So you can have defects in mitochondrial transport. Okay, any one of the transporters that carry these molecules inside the mitochondrial matrix. Okay, or problems in substrate utilization, primarily mutations in pyruvate dehydrogenase. This is the enzyme that is involved in converting pyruvate into acetyl coenzyme A. And it is really devastating pathology or disease if there is no pyruvate dehydrogenase functioning. The thing is, pyruvate would accumulate. Instead of being converted to acetyl coenzyme A, pyruvate would be converted to lactate, and that would cause metabolic acidosis. Okay, and there would be elevated lactate in blood as well as cerebrospinal fluid, as well as elevation in pyruvate and alanine in the system. There could be problems in any of the enzymes involved in the Krebs cycle. Okay, and in this case there would be excretion of large amounts of fumarate and succinate in urine, and that would cause mitochondrial encephalomyopathy. So there would be deficiency in the generation of ATP. Or you can have abnormalities in the respiratory chain reaction in any one of the 4 complexes, or in the oxidative phosphorylation. Okay, So the respiratory rate would be maximal in the absence of ADP, and that's an indication that respiratory control is lost. Okay, an example is Luff's disease or non-thyroidal hypermetabolism. So what would happen is that if there is a disconnection between the respiratory chain reaction and oxidative phosphorylation, there would be increased respiration, except that energy is exerted as heat rather than ATP. Now, you can also have genetic classification of mitochondrial diseases. You can have defects in the mitochondrial DNA itself in any one of the 13 subunits, for example. Okay, and these mutations are transmitted via maternal inheritance pattern. Okay, whereas and these are some examples of such diseases. There is the myoclonic epilepsy and ragged red fiber disease. Note that most of the affected organs are muscles, and to some extent the central nervous system. So there are different types of mutations or defects. You can have defect in the one of the tRNA molecules, and that are encoded by the mitochondrial genome, causing the MERRF condition. And there's another condition. There are other conditions, including Leber's hereditary optic neuropathy. Now, this condition here affects females more often than males. Inheritance pattern is again mitochondrial pattern rather than nuclear. And what you have in here is if deficiency of oxidative phosphorylation and ATP generation. So interestingly, the vision of individuals having this condition would be affected. Okay. And there are different types of mutations that can take place. Don't worry about the details, except just know that you have a problem in the respiratory chain reaction in here. Okay. Or mutations can arise as a result, or defects can arise as a result of mutations in the genomic DNA. And any one of the proteins, mitochondrial proteins, can be affected, and they follow the same pattern of inheritance that we know of: autosomal recessive, dominant, sex-linked, and so on. So let's talk about peroxisomes in general. So these are small membrane-enclosed organelles. Okay, and inside these organelles, you have a number of biochemical reactions that take place. Okay, and some feature of peroxisomes is that they can replicate by division. Now, the main proteins that make up peroxisomes are known as peroxins, and there are a number of these proteins. Most of them are metabolic enzymes, and they are synthesized mainly on free ribosomes, and then they are imported into peroxisomes. Okay, so these are the main functions of peroxisomes. They carry out oxidation reactions that can lead to the production of hydrogen peroxide. Hydrogen peroxide is harmful to cells, so they do also contain the enzyme catalase that converts hydrogen peroxide into water and oxygen. Okay, now there's also degradation or simplification of uric acid, amino acids, and fatty acids as well inside peroxisomes. Now note that fatty acids are oxidized in both peroxisomes and mitochondria as well. Interestingly, synthesis can also take place in peroxisomes. Synthesis of cholesterol, dolicol. We talked about dolicol before. which is the lipid carrier that is involved in end-linked glycosylation of proteins in the endoplasmic reticulum. There are other type of products produced by peroxisomes, including bile acids and the lipid plasmalogens. Now, how are peroxisomes formed? Basically, they are formed from the endoplasmic reticulum. Two different vesicles are formed carrying different proteins. These are called V1 and V2. Now what happens is that these two vesicles fuse with each other, forming a larger vesicle that also contain a channel protein known as importomer. So this importomer allows for the entry of cytosolic proteins or peroxisomal proteins synthesized in the cytosol inside peroxisomes. Okay, so what happens is that once they are synthesized, these peroxisomal proteins, they are recognized by a protein known as PEX5. So it's one of those proteins. This protein would be recognized by other proteins found on the surface of peroxisomes, and they can carry out, or they can transport the peroxisomal protein inside peroxisome. The PEX5 can be recycled, or it can be reused again, carrying another peroxisomal protein inside peroxisome. So I talked about how new peroxisomes are formed, and I said that they can arise from the endoplasmic reticulum. And another mechanism by which new peroxisomes can form is that they can grow and divide. So they can accumulate more proteins, and then eventually they can divide into two vesicles, or two peroxisomes, I'm sorry. Okay. So these peroxisomes undergo cycles of growth and division and so on. Now, there are a number of peroxisomal diseases, including that can result from defect in specific peroxisomal enzymes, mutations in any one of the PEX genes. An example is a disease known as Zellweger syndrome, which is a lethal disease that results in that can be caused by mutations in numerous peroxisomal proteins. Another example is X-linked adrenoleukodystrophy, where basically there is a defect in the fatty acid transporter or carrier that carries long chain fatty acids into peroxisomal membranes.